Hi everybody. Have you ever been to a clinic or a hospital and the doctor has prescribed you a really expensive medicine? If you wonder why it is highly priced, it simply is that this medicine has just been approved by FDA and released into the market. The main reason for its high cost is the huge amount of resources worth around 2 billion US dollars on average to spent usually by big pharmaceutical companies to produce a single medicine. Furthermore, the average time frame required to develop a medicine is between 12 to 15 years before it can be marketed. It's noteworthy that FDA approves only 50 new medicines per year, and this is due to its strict procedures and to make sure the medicine is safe and of high quality before it can be given to the patients. With the inception of the supercomputers era, in addition to the completion of the human genome map that had been made available in 2003, researchers in the pharmaceutical sector can save financial resources as well as the overall time that is taken for developing new medicines. As a result, this has effectively reduced the cost and time required, especially during the crucial times such as COVID-19 pandemic. Our project is to leverage natural product compounds synthesized from artificial intelligence technologies and to construct a big database which combine chemical structures and characteristics that resembles the chemical properties of synthetic medicines. The properties are tested using several artificial intelligence technologies to ensure that the virtually produced compounds strike the balance between natural products' diversity, uniqueness, and drug chemical properties. Furthermore, we screen these structures against the representative protein or enzyme of a specific disease such as colorectal cancer, depression, and Alzheimer's disease. Practically, this process involves several stages. Towards the end, select the best in-class molecules with a consensus of more than one model employing more than one screening method. The first stage, called QSAR screening method, involves building a prediction model based on biological data that have been recorded from real laboratory experiments, similar to the compounds we synthesized virtually. The second screening stage is called pharmacophore screening. In this type of screening, we map the connection points between the chemical compounds with the protein or enzyme of the targeted disease and draw a map for these connection points with the characteristics for each one of them. Later, we filter out all virtual structures using this map to select the best resulting structure. The final level of screening is called docking-based screening. In this step, we simulate the real attachment process between chemical structures and the disease-related protein or enzyme. Using advanced algorithms, we calculate the energy generated or required due to this connection and rank the virtual compounds based on these attachments. Eventually, we calculate the consensus score from the three screening processes and submit the best compounds to a more rigorous screening to check their stability inside the targeted protein. This process is called molecular dynamic simulation, which is handled by different algorithms that take into consideration temperature, atmospheric pressure, and other surrounding conditions. To examine the effectiveness of these screening methods, we synthesized one or more compounds from the highly ranked molecules produced by the consensus screening, in addition to the highly ranked ones from every single screening, and conduct proper biological assays to confirm the effectiveness of our method and ensure delivering lower priced medicines for the patients in the future. Thank you.